Let's say 21. Let's say 21.
That's literally the only time I could not. No, I could, yeah.
Oh, it's a 3. There's no.
No, but
That's so nice.
This work for everybody? Okay. So we're going to build on our discussion from last time and transition to something that's really quite remarkable that we in MSP. So we're going to talk about adaptive immunity. It's an important topic in this class. So the next couple of lectures, we're going to see that it is really a pretty expansive lecture, and I'm not going to plow through all of this in one day. We'll get to where we can in one day and kind of pick up where we left off on Thursday. So we're kind of trying to treat this week in this class as the immune system. And in the next week will be a lecture on experimental approaches that we use in bacterial pathogenesis, followed by our first foray into what pathogens are actually doing and some important components of what they do. And then we are going to wander into our first exam. So before that, you should have been able to see your first homework assignment. So it is a Google Doc that's available through Canvas. This is my first time dealing with Canvas and issuing assignments in this particular way, so I'm guessing we'll have a few hiccups. And I may shoot a couple of you an email. So I've noticed there are a few assignments that were submitted already, and I'm just curious if you're able to resubmit the assignment or kind of what you might need to do. Oh, you can?
Yeah. It just
all right. So it just kind of refreshes what's in there. Okay, that's good to know. This is my I've dealt with this. So
I wanted to know what it did before I hit the submit button. It was like, it's submitted, and I was like, oh shit.
Okay. All right. So it was kind of a.
There it goes.
Okay. So evidently, I will double check this, but you are welcome to submit as many times as you like before the deadline. The deadline is the day of the exam. And so the way that I view homework is this an important study tool. Many of the questions asked on homework will be similar to things that you would see on an exam. I would not say they are questions you will see on the exam. But questions in the same vein of things that I'm asking in homework are things that I expect you to be able to answer in a timely manner on an exam. So before we head into this wild and dense topic that I really don't do justice to, even though I'm giving 2 lectures, this topic. It's an incredibly fascinating, incredibly complex topic. But that said, we'll get where we can today. But before we head into today, we went over some pretty heavy stuff last time. Are there any questions last time? Okay. So the important thing to take away from last time is some of the players that we talked about last time. It's not like you'll never see them again. They actually are important players in adaptive immunity. We're going to see that there's a fair bit of crosstalk, especially the cellular mechanisms of innate and adaptive immune system components and the sort of very integral relationship that they have with each other. So, what am I going to cover today? I'm going to talk to you about what are these functional differences at a very high level, just reiterating again and again and again what are kind of contrasting things here. And where we're going to spend a lot of time in these next couple of lectures is talking about cells that participate in adaptive immunity, and mechanistically, what is their function and what are they doing? And we're going to kind of move through this slowly so that you're able to take these things in and kind of let them marinate, and you can ruminate on these things and digest what's going on. Because it's important that we build a good foundation here, because many of the mechanisms of what pathogens are doing inside of us and makes them successful is their ability to essentially bypass many of these mechanisms in a lot of different ways. And so, if I'm going to contrast at kind of a very categorical set of differences between the innate and adaptive immune systems that we'll really highlight in this next couple of lectures, contrasting to what we talked about last time, is that this adaptive immune system, this is something that's specific, it's found in vertebrates, and that this innate immune system, specifically this at the cellular level, this idea of toll-like receptors, and essentially pattern recognition, saying like this is what bacteria look like, and this is sort of what pathogens look like, and what I'm really doing is looking for big changes in abundance. These are something that invertebrates, vertebrates, and plants all do. And it's actually been one of the really remarkably interesting things, essentially across big, kind of chasms of differences between plants, insects, higher animals. This is a mechanism that's preserved using pattern recognition receptors, and the response is fast. And so this is one of the things that's very beneficial about this innate immune system. We'll see in contrast to the processes we're going to see over the next couple lectures, is that your adaptive immune system, the advantage is not in snappy response to new pathogens, because the things that happen, these processes we're going to talk about, is keep in mind that we're talking on order of days. We're not talking order of hours. Days or weeks, depending on the pathogen and various other factors. And what the innate immune system can sense, what it can see, that diversity is very small. So the slide from last time, where I kind of gave you a picture of a number of different toll-like receptors that we specifically have, and what we have kind of worked out are substrates that these particular TLRs will respond to. That is a seemingly very diverse, big list of things, but that diverse list really comes out of these receptors being fairly promiscuous in what they bind. So they are specific, but they're not really specific. They're looking for things that look like peptido-like structure, for example, or they look like LPS. But really what they're able to see, what our innate immune system, or basically any of our cells that are producing toll-like receptors, what they see is not very diverse. It's a fairly narrow set of substrates, and that the specificity is very broad. Importantly, there's not really what we call memory. And so if our innate immune system, if these toll-like receptors see Staph aureus versus seeing E. coli, like they're not really going to say like, man, I saw Staph aureus, and now I'm specifically equipped to deal with Staph aureus now in the future. And it importantly also exists prior to exposure. So this is sort of your frontline defense. It's there. It's basically there all the time. And like kind of any investment in things like this, the sort of broad thinking is that you want the sort of jack of all trades kind of system to deal with things first, and then you're going to come in and deal with things in a very specific manner at a later time. And that's the purview of this adaptive immune system. That essentially, just about anything that our bodies are exposed to that are not self, we're generally able to produce specific antibodies against those things, and we'll talk about how that happens. So as a result, what we can see with our adaptive immune system is incredibly specific. So we're able to differentiate different influenzas that we've been infected with from season to season. We're able to sense and detect differences in basically sub-species of Staph aureus that you may have been infected with. So you had a cut, a year ago, and you got a staph infection from one strain of Staph aureus. A year later, you get a cut, and you have a second staph infection that maybe is displaying a bunch of different antigens and is a different strain. We can develop specific immunity to two different, very specialized substrains of the same bacteria. And also importantly, it exists as a result of exposure. So we don't start just building a sea of different antibodies to everything. We have to see it in order for us to develop antibodies to something. And also that we have memory, that things that we've seen, and this is essentially the basis of vaccination, right? That we'll talk about that in a later lecture. But basically, you're building antigenic memory of something that your body sees something and stores the fact that it has seen this thing and developed antibodies to it to help you with later possible infections. So if I'm going to graphically describe that table, what goes on is there's a couple of major variables in terms of what our immune system is doing. And if I were to kind of categorically break them down, is how much of that microorganism is present. If we're thinking about viruses or bacteria or whatever that is not us, we've been infected with, how much of that thing is there? And how long have we been infected with that particular pathogen? So what we can start to think about is, say you get some infection, it's very local, I get a staph infection on my finger from a paper cut, and I essentially, I use bacitracin, and this red line here is essentially basically how much of this microorganism is present. I use bacitracin and I basically kill this sucker, this bug off that I have an infection with, within a day. Right? So this thing got kind of pus filled and gross. I put some Neosporin on it, wrapped it up, and a day later, infection's cleared up and I'm fine. So there are a couple of things that happen for me here. One is that what really begins to happen is that this gray area here, we can think of this as the domain of our innate immune system, where it's very effective. And it's very effective in dealing with low abundance of a particular microorganism in your body, and especially localized infections. And that basically, until you cross some threshold level, and this is, we'll come back to thinking about what defines this threshold later, but basically you can imagine there's some threshold that you need to cross for your adaptive immune system to actually be engaged, because it's an expensive process in terms of generating immunity. And so an infection has to proceed for quite a while, and also that this microorganism needs to really end up at fairly high titers before you're going to induce your adaptive immune response. And after you have generated immunity to a particular pathogen, your body is able to specifically go and find those pathogens and kill them. And as a result, you begin to clear this infection. And after this pathogen is gone, you establish what is known as immunological memory. and we're not going to get into the exact mechanistic steps of how this happens, but essentially a good number of the B cells that were producing antibodies, they basically transition into another kind of differentiated stage where they're continually producing antibodies against a particular thing, and they basically persist for years and years and years in the body. Whereas a lot of other lymphocytes that we're going to talk about, they're routinely cleared by your body. And so the players, when we're talking of some of the language that we're using when we talk about these specific defenses, is that the entire basis of this whole adaptive immune system is that we're developing immunity, we're developing essentially a specific response to a specific foreign antigen, and the language we're going to be using when we talk about these things are antigens are broadly, they're a foreign substance that's in your body. These are proteins, sugars, organic molecules. There's a number of inorganic things that we will generate immunity to as well. And epitopes, we're really talking about epitopes. These are really, we confine this discussion here to talking about the pieces of proteins that an antibody recognizes. And so shown here in this nice little diagram is a protein antigen that, say, a bacteria that's secreted. This might be a toxin, for example, and that what has happened is that our immune system has generated 3 different antibodies against 3 different epitopes of this same antigen. The antigen is kind of the big thing, and in our discussion in this class, we're really going to be focusing on proteins and sugars as what these antigens are. And the epitopes are essentially where what these antibodies are recognizing. And that importantly, an antigen is typically a kind of big macromolecule of some variety, and that we're essentially developing antibodies to lots of different epitopes of this particular antigen. Okay, so there's antigen, and then epitopes are basically these subset binding sites on a particular antigen by antibodies. And then we talk about the immunogenicity, or whether something is immunogenic, and that these just basically get into properties of how well are specific antigens basically inducing an adaptive immune response. So not all antigens are created equal. And so, for example, if I want to develop antibodies for some protein that I'm studying in my lab, I want to have antibodies so I can do Western blots and ELISAs and precipitation assays and all kinds of cool molecular biology experiments. And one of the things that I will need to do if I want to use antibodies to do that is generate the antibodies using a vertebrate host. So the most common thing that I would do is produce a bunch of my protein of interest. I'll send that off to a company where they will inject massive amounts of this protein into a mouse. And essentially that mouse is now exposed to this antigen, which is my protein that I'm interested in. Their adaptive immune system will generate a whole bunch of different antibodies to this protein. And different proteins that I would inject a mouse with have different immunogenic properties. So some proteins, you'll often hear these terms in kind of antibody generation in a molecular biology lab, is this is a really good antibody. Or we were able to generate a really good antibody against my protein of interest. And what this often has to do with is how immunogenic your protein is. So some proteins are not terribly immunogenic, and so you have to inject tons of this protein into a mouse over and over and over and maybe you're going to generate enough antibodies against enough different epitopes on this protein that you can attack it using, say, lectin. And alternatively, one thing that's immunogenic in mice might not be very immunogenic in us, but it might be, like extremely immunogenic in a goat, but be completely, non-immunogenic in a rabbit. And so this is why you end up seeing a lot of different animal hosts used to generate antibodies commercially. So that said, antigen, kind of a big umbrella. We're talking about proteins that are they're showing up in our body that they're not supposed to be there. Epitopes are essentially what our immune system has decided are good things to generate antibodies against. And often antigens will have multiple epitopes that your immune system has developed antibodies against to recognize. And then not all proteins are created equal. They have different immunogenic properties. So some proteins are better at eliciting an immune response, so we're really good at generating antibodies against specific epitopes against a particular protein that is very, very specific. Whereas some proteins are not good at doing this. And when we start thinking about pathogens and evading the immune system, if you cover yourself with proteins that are not very immunogenic, that's an advantageous thing. So, if I'm covering myself with something that this host that I'm infecting, that their immune system they're going to see it, and they're sure as heck going to try and generate antibodies against specific epitopes, but that those antibodies don't work very well against me because I'm covered with this protein or polysaccharide. Basically, modulating how immunogenic you are as a pathogen is an important trait for pathogens. Okay, so are there any questions so far? Yes?
Immunogenic in this case is just referring to the body's response level, not necessarily negative or positive, right? As far as getting rid of the bacteria. It's just a response modulation, right?
Yes.
It's not necessarily like its willingness to attack it or constrain it.
Yep. It's just getting at this idea of how easy is something to recognize versus saying protein A versus protein B. So protein A might be way easier for your immune system to generate specific adaptive immunity to it, see it, versus protein B. Yep. Good question. Thank you. Any other questions? Okay. So we're going to head into some of the meat of this discussion, and this is where things are going to get harder or not, depending on what your background is. Talking about the cells that participate in this specific defense. So when you were going back here and we're basically inducing our adaptive immune response, who are the players that are leading to this, that allow us to essentially clear pathogens and that these things don't just kill us every single time? And so some of these we've seen last time. So we talked a little bit about macrophages and dendritic cells and B lymphocytes. Importantly, what these are in our conversation today is that they're antigen-presenting cells. And this will become a bit clearer as to what I'm talking about. But there are specific types of cells that are actively looking to essentially grind up things that look fishy and will present antigens to our adaptive immune cells. And our adaptive immune cells will decide, based on what's presented to them, like, do I recognize this as something that's wrong? And if so, what happens next? So antigen-presenting cells are a very important class of cells. talk about B lymphocytes, and so whether these are plasma B cells or memory B cells, and essentially these are the antibody producers. And T lymphocytes, whether they be cytotoxic or helper. We'll talk about Th1 and Th2, T helper 1 and T helper 2 cells, cytotoxic T cells here. There's, in both of these lists gets much, much longer, much more complicated. One slide I'll show you, there's another type of T cell that we're not going to talk about contextually in this course, and I really want you to keep kind of a very bare bones picture of what's going on, because this can get wildly complicated really, really fast. And I think focusing on the complexity and the nitty-gritty details of a lot of this gets away from what I want you to take away from future lectures. So I showed you this slide last time, and that a lot of what we talked about last time were things that were involved in this innate immune response, and that these were all derived from these myeloid precursor cells. And importantly, all the cells that we're going to be talking about, these are essentially differentiated bone marrow stem cells, and it's just basically where they mature and become their final differentiated cells. And so what we're going to talk a lot about today are cells that are derived from these lymphoid precursors, and whether or not they matured in our thymus, or whether they matured in bone marrow. And so thymus matured lymphoid cells are T cells, and we'll talk about helper and cytotoxic T cells. And these bone marrow matured cells are B cells that once activated will become plasma cells that are actively producing secreted antibodies that are one of the pieces in our innate immune response that we talked about, or non-specific immune response that we talked about last time. Okay, so lymphocytes. These migrate to secondary lymphoid tissues after maturation. So these primary tissues that they mature in are bone marrow and thymus. So bone marrow again is B cells, thymus, T cells, and these are actively moving through our lymphatic system throughout our body all the time. Now they're not just sort of equally dispersed throughout our lymphatic system, but they like to reside in secondary sites throughout our body, and these are lymph nodes, our spleen, and then mucosal associated lymphoid tissue. So this MALT, again, is what we're going to be talking about a lot when we're talking about gastrointestinal pathogens, because there's a ton of MALT in our intestines. And it's important that we have a ton of this, because as we're going to talk about when we start talking about pathogens, and especially this gastroenteric environment, is that there is a lot of communication happening, because this is, there's a ton of surface area for our body that we're exposing to whatever we're eating, right? So, we generally think what we're eating is pretty clean stuff. It's not always the case. So what we're constantly doing with MALT, especially in our GI system, is sampling. So we're sampling what's being eaten. We're constantly asking, our immune system is constantly asking, like, is this good or bad? Have I seen this before? If I haven't seen it, does it look like self? If it doesn't look like self, is that bad? Should I generate immunity to the things that I'm seeing in my gut, basically? And we'll see, a lot of this kind of playing out in some coming lectures. So, what we'll see are a number of mesenteric lymph nodes, and so some of the highest density lymph nodes in your body are actually along your small intestine. And basically what these are a pile of these B and T cells basically waiting to interact with antigen-presenting cells to initiate this immune response in a very rapid way. So your lymph nodes, these are awesome things in that antigen-presenting cells, should they show up to a lymph node, they're essentially awash in B and T cells that we're going to talk about, this sort of interplay that all this has. But lymph nodes are a great sort of spatial organization of both activating T cells and B cells to produce this adaptive immune response, and then basically storing a lot of these B cells so that they start secreting antibodies that end up in the lymph system. They can get brought into your circulatory system, so on and so forth, and then you're basically very efficiently concentrating the players that are part of this this adaptive immune response. Specifically, it's important because when we start thinking about what these T and B cells are recognizing, and that I've hinted at this idea that there is variability in what these receptors are seeing, and then if every single B cell, or T cell, for example, let's say sees something else, sees something different with their T cell receptors that we'll talk about, if they were just diffuse everywhere, initiating a very rapid response with T cells that are just kind of willy-nilly everywhere instead of being concentrated in one place, essentially slows down this adaptive immune response, whereas concentrating these interactive events between cells in contained areas increases the probability of seeing things with your adaptive immune system more efficiently. Okay, so these lymphocytes that we're going to talk about, and why we'll say lymphocytes, they're some of the most abundant cell types in your lymphatic system. And so T cells, these can account for 80% of circulating lymphocytes, and there are three major types. And the two types that we're going to focus on that are germane to this course are cytotoxic T cells and helper T cells. And there are also these suppressor T cells. Essentially, you can kind of think about these as kind of the brakes on basically this whole system getting activated indiscriminately. B cells, we'll talk about plasma cells, formation of plasma cells, and there are also natural killer cells that are players, and we'll talk about these as well. And that these are essentially circulating lymphocytes, they're surveillance cells, and they're basically looking to kill stuff. That's their name implies. So there are a couple of different specific adaptive defenses, and we're going to end up kind of reusing some terminology that we used last time. Before I head into this, are there any questions? Yep.
So all of those cells on the previous page are A and C cells?
So these, on this slide, the antigen-presenting cells are dendritic cells and macrophages. Yes. These two, and I'm drawing a blank on neutrophils. I don't think they are. Yes. So these two and B cells are also antigen-presenting cells. Okay. Any other questions? So how many of you guys feel like you've seen some of this before? So how many of those people with your hands up have also been in immunology already? Okay. So it's a very, very cursory overview of a semester-long complicated topic. And so I'm trying to highlight the major players, major important pieces that become important when we're talking about kind of classical bacterial pathogens. and what are the kind of major host pathogen interfaces that we know of to date. So, two specific types of adaptive defenses that emerge. One is the cellular response. This is really mediated by cytotoxic T cells and helper T cells. And essentially, infected host cells, these are recognized and destroyed, and this is primarily the purview of cytotoxic T cells. We'll kind of talk a little bit about what this mechanism is. And humoral, the humoral component of adaptive immunity. This is mediated by B cells and T helper cells. Specifically up here, this is really the Th1, this B cells and Th2 cells. And essentially this is this idea that we're generating antibodies to recognize specific antigens in blood and lymphatic, in blood and lymph fluids. And also mucosal fluids. So, before we get into this picture, I wanted to ask our first question for the day. That is this one. That you have had a local, and when I say local, it's just like on your finger, staph aureus infection on your hand from a bad mosquito bite. So you scratch this thing open. I don't know about anyone else's experience in St. Louis this summer. I haven't lived here that long, but this is by far the worst mosquito summer ever. I don't really understand why. I'm sure there's some more explanations as to why. But it's been a miserable summer in my backyard. And so I've been covered with mosquito bites. You scratch them, they get infected. It's terrible. But like my description earlier, you treat yourself with bacitracin ointment. Bacitracin is a fairly broad spectrum antibiotic, and you clear this infection right away. So out of curiosity, you test yourself for the development of Staph aureus antibodies. And so you basically draw a little bit of your blood, spin out the red blood cells, and you throw your blood at some various antigens for different strains of strep or staph. And you're looking for coagulation, and you don't detect it. You've generated immunity to any kind of current Staph aureus antigens. And so what's the most likely explanation of these three? A, the level of Staph aureus present was not sufficient to elicit an adaptive immune response. B, your infection was not long enough to elicit an adaptive immune response. And C, the antibiotic ointment prevented antibodies from being formed because it killed all the Staph aureus cells.
Give them the time.
So I think,
You know what else I don't get? The gray area did not change when I was using this person. So you're thinking the I'm going more towards what I see like so that's for sure.
It's just us. Also the apartments. That whole section.
okay, I'm going to go ahead and open up the response window. And we will. There we go.
I'm not going to disagree because we got it wrong.
Right.
I don't think
Okay. Is anyone yet to respond? Okay. Never got to how many people are enrolled in this course, but one day. Okay. Let's take a peek. All right. Good and split. I like it.
Okay, I don't know that.
Okay. So this is a bad question in one sense, and it doesn't have one right answer. It's a great question, and then it doesn't have one right answer. So let's start with the one that nobody picked. So this ointment that you used, it killed all of the bacteria, and for the sake of argument, let's say that like this infection got really bad. I mean, you had kind of red streaks up your hand, like this, your finger was throbbing. You did this, and this infection was cleared. And for the sake of argument, let's say that you did test positive. So one of the things to keep in mind is that like our immune system doesn't really care. These cellular mechanisms of immunity, it doesn't care whether or not this pathogen is alive or dead. Okay, and it's all about seeing the proteins that are there and that are predominantly proteins that are displayed, or polysaccharides or other extracellular components of prokaryotes that are displayed that can be seen from the outside world, and that this is something to keep in mind with, say, having a really, really nasty septic infection. You might get pumped full of antibiotics. Those pathogens are dead, but you're still having an enormous immunogenic response. Basically, you're having still an enormous immune response to all of these dead bacteria, because you're still seeing those things. Your immune system is still seeing pathogens, whether they're dead or they're alive. And this is also a key component of vaccination, right? That you don't need to have a pathogen that's alive to develop a good immunogenic response to a pathogen. And so we're left with the two that are split here. And what either of these are really getting at has to do with that first slide, right? So plotting how much of that microorganism is present and how long has it been there. And both of these get at those two variables, right? That you treated yourself and that you might not have had this pathogen around long enough for you to essentially generate an adaptive immune response to this pathogen, especially for a pathogen that's not terribly immunogenic. And also that you actively cleared this infection, arguably before you ever generated adaptive immunity to it. So there's kind of some competing things going on here. So both A and B are arguable, that you can argue a case for either one that they're right? That it's either time or that's the amount of this pathogen. And really these things are very intimately and integrally related things. It's difficult to just completely decouple how much and how long, because both are incredibly important parameters in developing adaptive immunity to something. So are there any questions? Yes?
If you were to create like a situation like in a lab where you specifically induce someone with the appropriate level of bacteria to initiate the response, it would be A, because the level is ultimately what causes it to happen, right?
Yes, both it being there and it would be.
But I mean, like if you took away the long enough part because you let's say you injected someone with enough that it was like a certain level.
Yeah, if you were able to kind of like put it in and then remove it.
Right, so you take away the growth process of it, developing in your body, or does it not have to do with the infection itself growing in your body and more like how long your body has been exposed to it? When you say long enough.
So it's as I think we're going to see with some of the mechanistic details, it's really difficult to imagine being pulled apart. But
I guess I'm just confused, like what the previous slide that had the graph when it's talking about like length of infection, is that specifically referring to like what that microorganism is doing in the body and how it's like growing and taking over these different areas, or is it referring to your body reacting to the microorganism?
So that red line on that graph is how much of that microorganism. And so you're doing what you can to like curb the growth of this thing, but it's still an active infection. And once you've really seen this pathogen, developed antibodies against it, and you have a full-on immunological response to this pathogen, you'll start clearing this pathogen. So essentially specifically going and finding these pathogens, whether they're viruses or bacteria. Okay. So back to back to our picture at hand, where we left off before our break, is this. And that I talked a bit about antigen-presenting cells and these guys being really important for developing adaptive immunity. Indeed, they are. And there's a couple of things that I want to differentiate, and we'll kind of go through the bits and pieces of this about as far as we can today, and then I think we'll kind of, and I'm going to kind of deliberately go through this bit slowly, that I want to be able to kind of come back to a bunch of this again next time, and then kind of finish our discussion. But the things that we're going to contrast here are two different processes that are being driven by either antigen-presenting cells, and that these are using presenting antigens to T helper cells, eliciting a couple of different responses, versus the processes mediated by nucleated cells. So when we just talk about nucleated cells, these are not naturally antigen-presenting cells. They're operating with a very different process, in that there is an antigen presentation response that nucleated cells will engage in if they've been infected with a pathogen, for example, and that this is the purview of a different family of T cells, these cytotoxic T cells, and that it's a different specific programmed immune response in contrast to what happens, say, with this B cell activation and generation of antibodies. Essentially, cytotoxic T cells also have T cell receptors that we'll see are variable in what they look for. But if these cytotoxic T cells are activated, they basically use their receptor to go find specific things and kill them. Namely, other nucleated cells that have been infected by a particular pathogen. So, that said, we'll see if we get to it today, but kind of this particular pathway of activating your adaptive immune system has gotten a lot of traction recently in basically, like, if you can program this activation event by cytotoxic T cells, you can use cytotoxic T cells to target specific cell types in your body that are infected with specific viruses or are cancerous. So they're basically displaying the wrong surface antigens, for example, that are indicative of a particular cancerous tissue. So this programmed cytotoxic response, when we're talking about cytotoxic, these T cells basically go and hunt down our own cells that are presenting or have surface antigens that correspond to the pathogen that they're infected with. These cytotoxic T cells will go and find those infected host cells, our cells, and kill them. Essentially destroy the home that these pathogens have been residing. So, where we're going to spend a lot of time talking about, however, is this antigen-presenting cell response, and that this is, by and large, what sort of the run-of-the-mill response that we have to most bacterial pathogens, in that most bacterial pathogens are extracellular, that our antigen-presenting cells are going to go find them, grind them up, present them to our T helper cells, and elicit responses that result in cytokine production. So we talked about this last time. So basically, these immune activating chemicals, peptides. T helper 2 cells are also going to activate. They're going to activate B cells, which are going to result in antibody production. It's sort of kind of a daisy chain set of activation events. And the process of opsonization that we talked about last time, that is something that antigen-presenting cells like to do because they're naturally phagocytic, whereas nucleated cells are not naturally phagocytic, for example. Okay. So antigen presentation, the types of receptors that present foreign antigens from bacteria on host cells. So back in this slide here, I talked about MHC1, MHC2. What these are is the major histocompatibility complex, MHC, and as the last figure describes, there's two varieties. And there's a separate antigen presentation system, CD1, which anytime you see CD, when we're talking about our own human cells and saying like the CD1 receptor, what CD1 is talking about is cell differentiation. So it's a cell differentiation factor. And so we're basically generating antibodies against tons and tons of different surface proteins on tons and tons of cells and different tissues, and looking for signature surface receptors or surface proteins that let us, in terms of histology, say like, all right, this particular cell is this particular type because it's presenting CD1, CD38, CD35, and CD, whatever. And essentially just a categorical way of thinking about cell differentiation. And so CD1 is a surface protein, and it's actually responsible for presenting lipids to T cells. So importantly is this major histocompatibility complex. So class 1, MHC class 1 receptors, what this is a big deal for are intracellular pathogens. And it's one of the mechanisms that, especially when we start talking about intracellular pathogens, when I'm talking about intracellular pathogens, these are pathogens that actively invade host cells. And so after our first exam, we're really going to get into the nuts and bolts of what that process is. But if a nucleated cell, say an epithelial cell, is invaded by a bacterium, there on those epithelial cells are these MHC class 1 receptors, and what happens to an infected epithelial cell, if it doesn't just outright kill itself, is it will try and turn that vacuole that we'll see an intracellular pathogen is going to use to invade that epithelial cell. That formation of a lysosome, grinding up that pathogen, killing it. What is going to happen after that is that a lot of the peptides that are produced during that grinding up process will be basically presented on class 1 MHCs in these cells, and that's what those cytotoxic T cells are going to be looking for. To say like, all right, we have epithelial cells that are infected with this particular type of pathogen, and those cytotoxic T cells are going to be on the lookout for host cells that have been infected with this same pathogen. So the more pertinent to generation of antibodies is MHC class 2 receptors, and that this is really how we generate immunity to extracellular pathogens, or when pathogens are in their extracellular state, and that these are only found on antigen-presenting cells. And so these macrophages and other dendritic cells that are moving around, and they're basically gobbling things up. Part of that process of kind of indiscriminately gobbling things up, especially things that the toll-like receptors on those cells are binding to, is that basically these things that are gobbled up by macrophages, the fate of what gets gobbled up and put into vacuoles and ground up from the last lecture that we saw, lysosomal fusion, and basically breaking down whatever's in that vacuole. Things that are in that vacuole, they ultimately get presented on MHC class 2 receptors in antigen-presenting cells like macrophages, and that this is what's presented to T helper cells in places like lymph nodes. So, what does this process look like? So, what are MHC receptors? What are they doing? So, I've given a couple diagrams, and then we can kind of walk through this, and that so actual formation of these class 1 MHCs, This is triggered by viral or bacterial infection of nucleated cells. So things that are not antigen presenting cells. So they're not, other than those cell types, say epithelial cells can be induced to engage in phagocytosis, or endocytosis, so basically forming a vacuole and bringing something into the cell as a vacuole. That's not something that a lot of cells in our body do. And so epithelial cells, for example, are something that do not normally do this. Now, certain pathogens we'll see are induce this response. And when that happens inappropriately, that is something, that formation of a vacuole in these nucleated cell types that don't normally undergo endocytosis all the time, that is a very strong signal that something wrong has just happened. And that essentially you'll end up with formation of, similar to what happens in antigen-presenting cells, you'll end up with formation of a lysosome, and these peptides get ground up, or these pathogens get ground up, and essentially then you'll end up with the presence of abnormal peptides, or things that should not be in that type of cell in the cell, that these are then brought to ER, and in the ER is these class 1 MHC receptors. These mature in the Golgi, basically have vesicles that are formed that then allow transport of this MHC receptor to the cell membrane, and this is ultimately what gets presented outside of epithelial cells if they've been infected with a pathogen. And what this will then go do is interact with the T cell receptor that we're going to talk about probably not today on cytotoxic T cells. And we'll kind of get into the nitty-gritty of that next time. A bit more important for what we're wanting to talk about today is this canonical process that happens for phagocytic cells like macrophages and dendritic cells, is that they are going to basically take up a pathogen into a vacuole. Lysosomes in these phagocytic cells will fuse with these vacuoles. Lysosomal fusion results in degradation of pathogens, unless we'll see in some cases some pathogens are equipped with tools to deal with this environment. But by and large, most pathogens are going to, or bacteria in general, are going to die in here and be lysed, degraded, and that antigenic fragments. So unlike what happens with MHC1, class 2 MHC proteins, these are produced in ER. You end up with ER, basically vesicle fusion with these lysosomes, but then these MHC receptors are charged with peptides or other antigens, and then these are then displayed on the outside of these antigen-presenting cells. And so what ends up happening is that you basically grind these things up. MHCs are loaded with antigens from degradation of this bacteria or virus or pathogen, and that they're then displayed on the outside of these antigen-presenting cells. So antigen-presenting cells are constantly doing this, constantly displaying different MHC receptors, and what these then go do is that they allow for interaction with different types of T cells. And so MHC1, once charged with an antigen, this antigen-charged MHC1 receptor can now interact with a T cell receptor on a cytotoxic T cell, whereas the MHC2 that's charged with an antigen from a pathogen or other bacterium can now interact with T cell receptors, shown in gray, that are on T helper 1 and T helper 2 cells. Okay, so what I think we'll try and get through today is, all right, so these T cell receptors, what are these things doing? And these are receptors that are present on cytotoxic T cells, on on T helper cells, and that these are like antibodies, and that they also have a variable region. And so this is another example of where we're going to see that there is these big immunoglobulin proteins, a kind of broad family of proteins. But basically, they're going to be big blobby proteins that get stuck on the outside of these cells and on the outside of T cells. And what's most important about these proteins is that they have a variable region. And this variable region, shown here in red, what that does is, just like we talked about with antibodies, that the amino acid variants that exist in this particular region in T cells allow for recognition of different antigens. So it's important to keep in mind when we're thinking about these variable receptors is that every single T cell, unless it's basically emerged as a result of being there being a T cell activation event, and if these T cells are proliferated, is that every single T cell has a different variable region in this receptor. Okay? And so you can imagine there's a million T cells, and they all basically see a million different things. Okay? And that this is the result of genetic variation in this region of this gene producing these T cell receptors. So, which is also true for the variable regions of antibodies that we're going to talk about. So you have two sets of differentiated lymphoid cells. One matured in the thymus, the other matured in bone marrow. And that both of these populations have massive amounts of diversity in what these receptors see. Now, importantly, is that now we're talking about, what we're going to be talking about are a sort of daisy chain of events with T helper 2 cells. The requirement is that you basically have a population of, let's say, 1 million T cells that all respond to different things. And one of those million, maybe 2 of those million, are able to see this antigen presented by a macrophage that found a pathogen in your body. And that now, this one kind of 1 in a million event needs to go and search for its mate amongst another million random individuals in terms of B cells, and that you basically require this match of both seeing the same antigen for appropriate activation. And so what defines some of the specificity is that all of these T cells produce T cell receptors, but importantly, there are, again, different CD co-receptors on different T cell types. And so what dictates the specificity between MHC1 versus MHC2 recognition has to do with CD4 and CD8 co-receptors, respectively, on T cells or T helper cells. And this is one of the things that differentiates at sort of a histological level of like, all right, well, what defines a helper cell? A helper T cell, it's got a T cell receptor and has CD4 protein. And as we'll see next time, that basically there are, that in terms of T cell maturation, that there's sort of all T cells, and then T cells diverge into being CD4 positive or CD8 positive population T cells. Okay, and so the thing that I want to kind of end today on is how cytotoxic T cells recognize and infect or kill infected host cells. Let's say an epithelial cell has been infected by a intracellular pathogen like Salmonella. This epithelial cell has fended off Salmonella as best it could and was able to present some of Salmonella's surface proteins on a class 1 MHC receptor. And now this cytotoxic T cell has its T cell receptor that can, with its variable region, sees that antigen and says, yes, I see what you're presenting to me. And also has this CD8 co-receptor that lets it interact with the MHC1 receptor. And once this event happens, is that this T cell begins dumping out all sorts of toxic stuff that is going to kill this infected cell. Concomitantly, one of the things that happens with these cytotoxic T cells is that when they engage in this, and that they've recognized an antigen on an infected epithelial cell, let's say, and that it has produced a bunch of pore-forming proteins, killed off this infected cell, is that this target cell now died, but what's going to happen next is that not only was this T cell important for recognizing and killing off this pathogen, but the cytotoxic T cell is going to now proliferate. So it's activated itself and said, okay, I have found something bad, time for me to essentially expand my population of myself. So that now these cytotoxic T cells, it's not just one activated cytotoxic T cell, but it's going to give essentially birth to a large number of also activated cytotoxic T cells that are going to see the same antigen that this original cytotoxic T cell had. So the whole basis, where I'm going to end here in just a minute, there's a whole kind of thread of medical research now and like thinking about how can we reprogram this response. So if I were to harvest a bunch of T cells from your body and if we filter out all of the other T cells that I don't care about and just look at your cytotoxic T cell population, and I can activate some of those and select for ones that are going to be responsive to a particular virus or a specific cancer protein. That one of the results of cancerous cell development is that they often start producing inappropriate cell surface receptors. And that you can say, all right, well, I'm going to basically activate T cells, cytotoxic T cells from your own body to go and find that surface receptor on a cancer cell. And now I'm going to re-inject you with a large population of activated cytotoxic T cells that are basically programmed to go look for that particular cell surface receptor. So there's a lot of exciting things in this cytotoxic T cell research area, and we'll pick up our discussion next time.